So yeah, let's talk about DNA error repair. There was two main things that you guys had to know, was mismatch repair and then nucleotide excision repair. The polymerase are really, really good, right? Polymerase 3 is really, really fast. It's really efficient at putting down that template strand, or making that complementary strand, rather. However, because it's going so fast and laying down those nucleotides so quickly, it's entirely possible that some mistakes are made. And those we could also consider mutagens if there are physical agents or things that are in the environment that are actually causing that. So cigarette smoke causing damage to the lungs, for example, x-rays causing damage to all of your DNA. There can also just be some spontaneous mistakes that are made. So we're going to talk about two different kinds of errors that happen and then the repairs that follow to make sure that your DNA is fixed and maintained. So the first is mismatch repair. So this is when there's the wrong letter is across from the complementary strand. So if this was an A and then instead a G is put in, which would actually make it really, really big, the DNA can actually replicate or recognize that that mistake was being made. So first thing that happens, recognition of the damage. So noticing that A wasn't matched with T, but A was matched with G instead. The removal of that damaged region by something called a nuclease. And then we're going to see polymerase 2 just boop, 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 puts down the new nucleotides, and then ligase just seals it up together. So that's our first version of this. So on our image, if this was A and this was supposed to be a T, but instead it put a G in, you'll notice that that DNA got a lot bigger, for example. Then there's a complex that will actually recognize this, cut that out. Here's our little nuclease that cuts that out. And then polymerase will just come on along, put in a new strand, and then ligase will seal that up. So that's it, right? Second one, UV damage can cause something called thymine dimers, where two T's that are sitting next to each other, instead of being connected to two A's on the other side, instead, for some reason, because of UV damage, the T's actually connect with each other. And so UV damage can cause this problem. And so again, we're going to see a nuclease removes that piece that was damaged, and then we'll fix it. So here were two T's right next to each other, should be connected to A's on the other strand, but instead, for some reason, because of UV light, the two T's actually connect into each other. We call that a thymine dimer. And so we'll see this photolase, a specific kind of nuclease, will cut that up, and then we'll see it actually gets fixed up from there. And so those are the couple that you guys needed to know. What's important to remember is that these error-proofing measures are pretty darn good, and truly DNA polymerase 3 does a great job putting down the nucleotides in general. However, sometimes these get missed, and so then those changes actually become permanent and potentially could get passed down to the next generation. And so when we think about mutations being the original source of genetic variation, this is where that's happening, and that actually can be responsible for new species or adaptations, just depending on what those mutations are doing. And that was it. That's all I